Good morning, everyone, or good afternoon, or good evening, wherever you are. Um, my name is Christy Watson, and I am the Director of Research and Development here at Precision Quantomics. Thank you for joining us today for our webinar on advancing protein quantification and its role in drug metabolism and translational research. So as we get started, first, we're going to start with a brief overview of Precision Quantomics and then move on to our speaker's presentation. So at PQ, we use advanced LC-MS technologies to support pharmaceutical drug development by integrating quantitative proteomics data into new approach methodologies, including lab-on-a-chip, DMPK, IVIVE, and PBPK modeling. Our solutions include specialized products, testing services, and the TP-Atlas, which is our state-of-the-art database designed to support comprehensive PBPK modeling with high-quality proteomics data, including data from underrepresented populations. So now on to our speakers. Our first speaker is Dr. Tina Mueller. She is a scientific advisor for ADMET services at BioIVT, where she supports drug discovery and development programs through expertise in enzyme-mediated drug metabolism, ADME tox workflows, and translational in vitro systems. She earned her PhD in biochemistry from the Swiss Federation Institute of Technology, ETH Zurich, and brings more than 20 years of experience spanning biochemistry, pharmacology, and preclinical drug development. Dr. Mueller has contributed extensively to advancing in vitro metabolism and translational ADME strategies, including metabolic stability studies, protein degrader characterization, and species comparison workflows. She frequently presents on practical approaches for improving preclinical decision-making and optimizing translational relevance in drug development programs. The second part of our talk will be given by Dr. Joanna Barbara. She is a general manager of ADME services at BioIVT, where she leads scientific operations and strategy supporting in vitro drug metabolism and translational ADME research. She obtained her PhD in analytical chemistry from the University of Florida and has spent more than a decade advancing bioanalytical and drug metabolism capabilities across the pharmaceutical research industry. Prior to joining BioIVT, Dr. Barbara held scientific leadership roles at Xenotech, where she helped expand analytical and in vitro metabolism services focused on complex drug modalities and translational research applications. Her expertise includes mass spectrometry, metabolite profiling, transporter research, peptide therapeutics, and in vitro metabolism systems supporting drug discovery and development. All right, so now I will turn things over to Dr. Mueller and Dr. Barbara for today's webinar.
Yeah, thank you so much, Christy, for the nice introduction. And hello, everyone, and thank you for joining. As Christy said, I'm Tina Mueller, and I'm going to speak today together with Joanna Barbara, who will do the second part. And we're talking about evaluating DDI risk with a focus on practical in vitro ADME strategies. And then Joanna will take over and do a deep dive on peptide metabolism. I hope you enjoy, and I'm looking forward to your questions at the end. So let me just start by outlining what I'll cover in the first part. I'll start with why drug-drug interaction or DDI matters, and then briefly review how we assess DDI risk in vitro. From there, I'll highlight three key trends shaping ADME today. The first one is a focus on non-CYP enzyme metabolism, then a trend towards low turnover compounds, and last but not least, how do we need to adapt and modify ADME strategies to new modalities? And as mentioned before, I will then hand it over to Joanna, who will walk us through exciting new data from a peptide metabolism case study. So let's begin with why DDI assessment is so critical. And let me ask you a very simple question to start us off. Have you taken a prescription drug in the last month? Or actually, have you taken more than one? And no worries, you don't have to answer that in public. But if I had to guess, many of you answered that question with a yes. And that yes or that guess is based on statistics. Looking at numbers from the CDC, in 2021, almost 65% of adults took at least one prescription drug in the last year. And the number of people who took 5 or more drugs in just the last 30 days was over 10%. And for adults older than 65, that number was as high as 43%. So why does that matter? Drugs are predominantly metabolized by the same groups of enzymes. And that means that the presence of one drug can influence the behavior of another drug. So let's look at an example where one drug changes the fate of another drug. And the example here is mibafadril, also known under the brand name Posicor. So mibafadril is a selective calcium blocker that was approved in 1997 and used to treat hypertension and chronic angina pectoris. It is a strong metabolism-dependent inhibitor of CYP3A4, and if taken simultaneously with statins, such as, for instance, simvastatin, the systematic exposure to the statin increases quite dramatically as the drug is not metabolized as expected, and the risk of toxicity increases. Indeed, the combination led to the breakdown of skeletal muscle and acute renal failure, and ultimately resulted in the withdrawal of Posicor from the market in 1998. So with this example of drug-drug interactions in mind, what are the possible effects of drug-drug interactions? So the figure here on the left illustrates that in a very simple way. When a drug is taken at the same time as another drug, basically 3 things can happen. The drug is cleared faster from the body, which can result in lower therapeutic effects. In contrast, as shown here in the middle, the therapeutic effects could be higher or last longer if the drug is not metabolized. And last, the drug could cause adverse effects. And to summarize, any of these outcomes obviously might be undesired. Now, to understand the risk for these DDI, we need to understand ADME characteristics of the compounds. And understanding these characteristics is also crucial or especially crucial when we develop new drugs, as the obvious goal is to create a compound that has a therapeutic benefit and can be dosed to a patient safely. So in other words, the drug candidates and the ultimate drug should have desirable ADME properties. Which brings us to in vitro ADME studies. So these are designed to evaluate the ADME properties of drug candidates, and they answer questions such as how stable is a drug, or what enzymes are involved in the metabolism, or is the compound a substrate of a transporter, to just name a few. So the results of these studies are then also used to predict the risk for drug-drug interactions and inform whether clinical DDI studies are needed. And last but not least, conducting these studies is needed to be compliant with regulatory guidance. Shown here is the ICH M12, or the ICH Drug Interaction Studies M12, which was finalized in May 2024 and replaces most local guidelines such as the FDA DDI guidance. So let's now take a quick look at the studies that are part of in vitro ADME evaluations. Generally speaking, we have 3 main areas. First, we have drug metabolism, then drug metabolizing enzymes, and last but not least, drug transporters. So for drug metabolism, there are 3 main study types. We have metabolic stability, and that study assesses the in vitro clearance of a drug candidate. Then in metabolite profiling and identification, that's a study, as the name suggests, that profiles metabolites and can also propose biotransformation pathways of drug candidates. And last, we have reaction phenotyping. These studies identifies the enzymes, CYP, as well as some non-CYP enzymes that are involved in the metabolism of drug candidates. Now, for drug metabolizing enzymes, let's focus today on CYP enzymes. So we will focus on CYP inhibition and CYP induction which evaluate the potential to inhibit or induce CYP enzymes respectively. And then last, in drug transporter studies, the drug candidates are evaluated for their potential of being either a substrate or an inhibitor of the transporters. So now let's switch gears and talk about the 3 different trends in drug development and how they affect the in vitro ADME space. First, I would like to introduce the growing interest in non-CYP enzyme pathways. So let's first look at approved drugs and what enzymes metabolize these molecules. Here in this pie chart is an analysis from Matt Cerny's paper from 2016. And you can see that in all 3 time periods from 2006 to 2015, approximately 50% or more of drugs are primarily metabolized by CYP enzymes, which is shown in the red slices. So despite increasing awareness, and what I also hear when talking to drug developers in the field, CYP enzymes continue to play the central role in drug metabolism. Now, the fact that most approved drugs are being metabolized by CYP enzymes means that these drugs are inherently at higher risk for drug-drug interactions, since CYP enzymes are frequently inhibited or induced by co-administered compounds. So we could state that to reduce the risk of DDI, drugs should be designed that are metabolized by non-CYP enzymes, which leads to our central hypothesis. If developers are actively trying to reduce CYP involvement, then we would expect to see a growing emphasis on non-CYP enzyme evaluations and an increased contribution of those pathways in preclinical studies over time. So to address this hypothesis, we evaluated 393 reaction phenotyping reports at BioIVT and Xenotech from 2000 to 2023, and we recorded what enzymes were involved or contributed to metabolism, as was concluded in the reports in the results and discussion section of the studies. So here are the results shown as pie charts again, and in green we have CYP enzymes, in blue non-CYP, and then in yellows are studies that found both of those enzyme classes to be involved. And you can see that the CYP enzymes account for about 70 to 73% throughout all 3 time periods. So on the left, we have 2000 to 2007. Then we have 2008 to 2015 and 2016 to 2023. So in other words, non-CYP CYP enzymes account for about 70% in all of those 3 time periods. And non-CYP enzymes contribute just in a minority of cases. And actually, it is remarkably stable over those 3 time periods. So overall, despite an increased awareness of CYP-mediated DDI risk, we do not observe a shift toward non-CYP enzyme metabolism in preclinical reaction phenotyping studies. I also wanted to mention here that the relative contributions of CYP and non-CYP enzymes in preclinical candidates actually closely mirrors what we see in approved drugs. And this also suggests that, at least to date, if there is a strategic intent to reduce CYP involvement, it has not yet translated into a measurable change in preclinical drug development studies. So now let's move on to the next trend, a shift or a desire to design low turnover compounds. Now, this is interesting for various reasons, such as to extend drug exposure or reduce dosing frequency, or allowing lower dosages, to just name a few. And as an example of that trend, a survey of Pfizer's internal pipeline in 2015 showed that approximately 30% of compounds reported no measurable turnover during standard incubation times, clearly reflecting the importance of metabolically stable molecules in the pipeline. So we were interested whether we can see a growing contribution of stable compounds, and our hypothesis was that over time, a shift towards compounds with lower turnover can be observed in preclinical drug candidates. Now, the method was very similar to the previous analysis, but we extended reaction phenotyping to also include metabolic stability studies. Again, these were stability studies which were conducted at Sinotech and BioIVT in the same time period. With that inclusion, I also want to mention that we included an additional test system. So we had data from human liver microsomes as well as primary human hepatocytes. And the data was fitted to single exponential decay, and then intrinsic clearance was calculated, and metabolic turnover categories were defined, as you will see on the next slide. So this is the results of the intrinsic clearance study covering the time period from 2000 to 2023. In these first two pie charts, we have metabolic stability studies in human liver microsomes. And we do see a shift after 2015 toward lower intrinsic clearance, shown in the two darker blues in the pie charts. And that actually goes hand in hand with a decrease in high turnover compounds, which are shown in the lightest blue. Now, when we look at primary human hepatocytes, we see a similar trend where the fraction of stable compounds in the darkest green increases from about half to roughly 2/3. Also suggesting a shift toward more metabolically stable molecules. Now, last, when we look at reaction phenotyping data back to human liver microsomes, we observe an even more pronounced increase of low and very low intrinsic clearance compounds, as seen in the purple and the darkest pink in that pie chart there over time. So overall, these data indicate a sustained shift toward low turnover compounds across all different test systems, and we indeed see the strongest effect in reaction phenotyping. And we attribute this strongest effect in reaction phenotyping to the fact that these studies are actually being conducted later in development. So drug developers have already selected for low turnover compounds, further confirming the desire to develop more stable compounds. And now, of course, working for BioIVT, I need to bring up here that this highlights the growing need for tools to assess metabolism of low clearance compounds, with obviously the choice here would be hepatopac. So now last, let's look at the third trend. Expansion beyond traditional small molecules and how ADME is adapting, or in vitro ADME studies. So small molecules still dominate the FDA approvals and development pipelines, as reflected here in the chart on the right, which represents the majority, which represents the approvals by modality in 2025 at CDER. However, the fastest area of innovation is clearly shifting toward more complex modalities. Now, these include antibody drug candidates, so ADCs, oligonucleotides, protein degraders, peptides, as well as radiopharmaceuticals. And each of these new modalities brings distinct ADME and DDI evaluation challenges. And I want to mention here that the question is not necessarily whether typical small molecule in vitro ADME studies are needed, but rather which assays are most relevant and how experimental conditions should and must be adapted to properly assess these emerging modalities. So let's look at the different types, and let's start with protein degraders, which are dear to my heart, and what it takes to adapt traditional small molecule ADME approaches to these beyond rule of 5 compounds. The overall motto here is to evaluate analogous to small molecules. And I'm not going to go over all of these again, but just highlight a couple of differences that need to be adapted, such as assay conditions and maybe also timelines. So let me just call out some of these differences, starting with the physicochemical properties. Protein degraders, and here I'm focusing on PROTACs, so heterobifunctional protein degraders. They are larger, they are more flexible, and they have higher polar surfaces, areas, and also multiple rotatable bonds. Now, with that said, they are often administered orally, and despite showing limited absorption and oral bioavailability. And last, I would like to mention that they have complex metabolism because they are larger and they contain multiple soft spots. So these differences directly translate into the challenges listed on the right. So early on in development or in discovery, we see issues with solubility, stability, and non-specific binding. As an example, it has been reported, and actually we have seen that as well internally, that many protein degraders are unstable in PBS. Then cell permeability is also often limited and critical to evaluate early in development. Unfortunately, traditional small molecule approaches such as KCO2 permeability assays have shown to be poor predictors of in vivo cell permeability as well as oral absorption. And that's definitely an area where further technical development is needed. Then high protein binding might be a challenge, and evaluation should be prioritized early in development. And last, complex metabolic pathways make both stability assays and MetID more challenging and requires expertise and appropriate bioanalytical capabilities. Next, let's talk a little bit about ADCs. And as a key starting point, I would like to specifically call out that the ICH-M12 guidance mentions that both components, so the antibody as well as the small molecule payload, need to be considered in the evaluation. Now, to mention this upfront, the DDI risk for the antibody is generally considered low. And in addition, the systemic exposure of the released payload is also often low, which often limits its potential to act as a perpetrator. So it is more likely to be a victim or an object. However, the payload, and in some cases the linker, should be evaluated similarly to small molecules. And then beyond typical DDI studies or in vitro ADME evaluations, linker stability and cleavage need to be characterized across biologically relevant matrices, such as plasma, lysosomes, and maybe even tumor subcellular fractions, to understand where and how payload is released and what effect metabolites have. And finally, I would like to mention the potential for immune-mediated effects. So those can be assessed in vitro using cytokine release assays, as well as indirect suppression of CYP enzymes in addition, which can also influence DDI risk. Moving on to oligonucleotide therapeutics, such as siRNA and ASOs, I want to state up front here that the DDI risk for oligonucleotides is beyond the scope of the ICH-M12 guidance, and the FDA finalized the clinical pharmacology considerations for the development of oligonucleotide therapeutics in 2024. With that said, oligonucleotides are still often considered small molecules and are often evaluated accordingly. But of course, the strategy needs to be adapted to reflect the distinct biology and very also often the backbone and the modifications in the actual molecule. Now, starting with metabolism, traditional metabolite profiling and reaction phenotyping are generally less relevant for obvious reasons, as oligonucleotides are primarily degraded by nucleases rather than CYP enzymes. Instead, it becomes more important to evaluate stability in biologically relevant systems such as, for instance, plasma and lysosomes. Now, from a DDI perspective, CYP-mediated risk is typically considered low. Again, though, depending on the program, targeted assessment may still be required, and indeed, regulatory guidance actually emphasizes the need for a clear scientific justification if these studies are not performed. Similarly, transporter-based interactions are generally minimal for oligonucleotides, both as substrates and inhibitors. And then plasma protein binding can be significant, as oligonucleotides can be highly protein bound, specifically for ASOs. Now, again, moving beyond the most common in vitro assays and to better understand both on and off target effects, more advanced systems, such as long-term hepatocyte co-culture models like hepatopac, can also be very valuable. And finally, as with other emerging modalities, the immune-mediated effects should be considered. So last but not least, peptides. Overall, peptides tend to present a lower risk for typical small molecule drug-drug interactions. But of course, it is still important to perform a mechanism-based risk-driven assessment. And I just would like to briefly cover here metabolism, as peptides are primarily degraded by proteolytic enzymes, so rather than by cytochrome P450 enzymes. But as Joanna will go into more details, I really want to call out that other test systems might also be important, such as S9 and lysosomes. And with that, I would like to finish with a few key takeaways. First, in vitro ADME and DDI studies remain essential. These studies are critical for understanding drug disposition, potential drug-drug interactions, and overall safety risks. And they really form the foundation of any drug development program. Second, we have CYP-mediated metabolism. It continues to dominate, particularly for small molecules. At the same time, we are seeing an increasing number of low turnover compounds, which can be more challenging to characterize, and test systems need to be carefully evaluated and selected. And finally, as we move beyond small molecules, ADME strategies need to evolve. Standard assays and approaches we have historically used are not always directly applicable, so we need to adjust and modify as needed. And with that, I would like to mention a couple of people who have helped in all of those endeavors. Brian Ogilvy, Courtney Noah, as well as Ken Christstaff, are colleagues of mine, scientific affairs team. And then Lois Haupt and Chris Ball have contributed to the analysis studies, and then Andrew Taylor, who is not with BioIVT anymore, as well. If you have any questions, let me know. I'm happy to talk about this. And with that, I'm going to hand it over to Joanna.
Thank you very much, Tina. As Tina said, I'm going to dive a little bit deeper into some case studies and data that we've been working on to represent some of the things she was talking about with respect to the peptides. Although, as noted, the cytochrome P450 enzymes are still really key to the pipeline overall, the pipeline is evolving. It's just not as marked as people would expect to see necessarily. So over the past 10 years, we've seen a decrease in the small molecule development, which would implicate the cytochrome P450s by about 25%. And we've seen a total growth, at least 5% each, across other new modalities. So cell gene therapy, RNAs, ADCs, peptides, all the things that Tina was just talking about are all growing. Because of that, it means that the traditional toolkit that we're all used to using in vitro doesn't really apply in the same way that it used to. It becomes a little bit less relevant. So we need to think about the tools in a different way and take some different approaches to be able to develop these drugs and assess liability, safety, stability, all of the things that we are used to looking at as ADME scientists. So because of that, in vitro test systems are in a little bit of a different place in the world. We're in a new paradigm, is how I like to put it. If we think about the metabolism of larger molecules or biomolecules versus small molecules, you can see that there are some very different considerations to take into account. When we're looking at uptake in small molecules, we're usually talking a lot about transport or active transport and passive permeability. In contrast, with biomolecules, we have to be thinking more about things like endocytosis and phagocytosis. We're no longer just looking at the traditional drug metabolizing tissues, either liver, intestine, and kidney are the big three when you're talking about small molecules. But if you start thinking about the biomolecules, we're looking at all kinds of tissues throughout the body. Honestly, most tissues in the human body. The metabolism is no longer by our traditional drug metabolizing enzymes, CYPs, UGTs, salts with pHs around 7.4 for optimal activity. We're looking more at degradation pathway enzymes, the catabolism enzymes, nucleases, peptidases, and we're thinking about pH 4.4 to 5 for the lysosomal compartment, which we'll talk about in more detail presently. Even the subcellular components we're thinking about are a little bit different. We're all very used to doing experiments on microsomes and cytosol and S9, but now we have to think more about things like the lysosomal and peroxisomal fraction than we had to before. Whole cells remain appropriate for both, it just depends on what you're actually looking at. So we did some work with some GLP-1 receptor agonists, and we specifically chose these drugs that we were looking at, which are the yellow highlighted ones, because we were looking at things that either currently are or have the potential to be dosed to a very large patient population, because in the longer run, we're interested in disease state and off-target effects. So we were looking at semaglutide and tirzepatide, which are widely prescribed at this point, and everybody knows them as Wegovy and Zepbound and things. And we also included orforglipron, which is a small molecule GLP-1. It's the newest addition to the family. It's only approved since April 1st of 2026. First one to be approved for obesity first, which is interesting. But in the longer run, we believe that that will be quite widely prescribed as well. So that was included in our study. And if you look at these drugs, they're very different. Semaglutide and tirzepatide are larger biomolecules. We'll talk a little bit more about the specific chemistries in a second, but in contrast, orforglipron is a small molecule, so we're covering very different chemistries when we're thinking about these drugs. All of these are based on GLP-1, and I like to take a little step back because we're going to talk about the drug design in a second, which is a native protein in our bodies, or peptide in our bodies, and it is very short-lived. Although it does generate all the effects that we're used to thinking about with GLP-1 receptor agonists, it's very short-lived because it's extremely rapidly catabolized by DPP-4 in the body. So it only hangs around for a minute or two, which means that all the effects that are caused are fairly short-lived. So in terms of turning that into a druggable compound, one of the goals was to make sure it was longer lived. And you can see that in the way that semaglutide is designed. It's quite clever. It's a clever molecule. So it's a native GLP-1 backbone, except there are a few substitutions. The first alanine is substituted with a non-natural amino acid, AIB. And the reason for that is it blocks the site of that DPP-4 metabolism. So it increases the stability of the peptide. Then if you look at the lysine at position 20, it's modified with a linker, a spacer, and then a fatty acid side chain. That has a very specific job too. That binds to albumin in our bodies. This is subcutaneously injected, and it sort of makes an in vivo long-term slow release effect. So it's stuck to this albumin, and over time, a little bit comes off and a little bit comes off. The spacer and the linker are designed to be just the right size to make sure the peptide's still available for activity. And then the final substitution here is that last lysine is substituted with an arginine, because otherwise you would end up with two side chains when you substituted the first lysine. Tirzepatide is a similar idea. It's a little bit more complex. It's based on GIP instead of GLP-1. So the peptide backbone is different. It's a little bit longer. And the yellow substitutions are where amino acids have been substituted either to reduce the likelihood of that DPP-4 liability, which I mentioned a minute ago, or to mimic GLP-1 so that it will fit into the GLP-1 receptor binding pocket as well as the GIP. Then the gray amino acid substitutions are about enhancing PK effects and things. and then a similar linker, spacer, and fatty diacid for the albumin binding and the extended release. So they're very cleverly designed. When we think about that, though, the fact is that now these compounds have multiple different chemistries in them, and we have to think about them in a different way than if we're just looking at one type of chemistry and one type of metabolic pathway. With small molecules, we're looking at non-catabolizing enzymes. These comprise some pieces of small molecules and some pieces of biomolecules, like the peptide and the fatty diacid. So the peptide backbone in pink is going to undergo peptide cleavage catabolism by peptidases, and that's predominantly in the lysosomes in the extracellular compartment and some tissue as well. And then the linker is fairly stable. It's not entirely stable because if you look at it, there's some amides. There's two, which are going to be susceptible to hydrolysis. Then the glutamic acid spacer is reasonably stable, but it is known to undergo some amide hydrolysis and redox metabolism. We were not sure where that was going to occur when we started out this experiment. And then the fatty diacid it undergoes beta oxidation fatty acid chain shortening, the very classic 2-carbon chain shortening reaction. And that occurs in peroxisomes when it's a long chain and mitochondria when it's a short chain. So there's a lot going on when it comes to metabolism of these. And these drugs are endocytosed, which means that they will ultimately accumulate in the lysosomes. So if you look on the left, you can see a nice cartoon of a GLP-1 actually modified with an estradiol receptor binding motif as well. And it's endocytosed into the cell. Then that endosome is going to fuse with the lysosomes and shuttle the receptor agonist from the endosome into the lysosome, where a lot of the catabolism will happen. Lysosomes are heterogeneous membrane-bound organelles. Their job is to get rid of biomolecules, though there's a lot of peptide and protein cleavage in there. Oligonucleotides, RNAs are degraded in there. They're all under an acidic environment, which is necessary for the optimal activity of the enzymes. One of the very high abundance enzymes in these lysosomes is the cathepsin family, which is proteases. Because of that acid environment and the membrane surrounding, there is also a sequestration effect that happens, but we're not going to discuss that very much today. It's just an important consideration when you're thinking about working with lysosomes. And a single hepatocyte contains about 250 of these. So more than you expect in the sort of traditional cartoons like on the left, where they're dotted about. They're actually fairly high density, and they don't occur on their own. They are located very closely in close proximity to other cell organelles. So you can see in the right side and in the micrograph, the lysosomes are adjacent to peroxisomes and mitochondria, which is why the shuttling of the fatty acid chain shortening can happen. So that there's some things to think about when we're trying to explore lysosomal metabolism now. When we started out looking at lysosomes, it was hard to get something that was high specific activity and high concentration in terms of the enzymes that we're interested in. And so about 10 years ago, a group of my colleagues determined a way to isolate a high specific activity, high purity fraction of lysosomes. And the way that they do that is they isolate the crude lysosomal fraction from fresh human liver tissue. You get a much better yield if you use fresh. Then you do some low-speed clarification homogenization to get this crude fraction. You purify it by a density gradient. It can be percol, sucrose, that sort of thing. And at that point, that's when the magic starts to happen. The problem with lysosomes when it comes to ultracentrifugation and trying to separate them out from the other organelles is that they're isopycnic. They have the same density as mitochondria. So it's really hard to split out the two. So for us, in our proof of concept work, we evaluated 12 fractions. You can see them here. And we took each of the fractions and we evaluated them for enzyme activity associated with the lysosomes, acid phosphatase, cathepsin, versus the mitochondria, cytochrome oxidase. And you can see that the highest lysosomal activities are occurring in the top fractions. So we would take those and use those as our lysosomal fraction. But the bottom fractions have activity more associated with mitochondria. And so we would discard that because that's the mitochondrial peroxisomal fraction. You can also do experiments in the whole lot if you're interested in the mitochondrial peroxisomal contributions. Just depends what you're trying to do. And then we took those lysosomes and we did a fairly simple incubation. We incubate at quite high concentrations, 2 micromolar for this. for a couple of reasons. One, we weren't sure how it was going to go. And two, we are ultimately interested in looking at the biotransformation of this. So we wanted to make sure there was plenty of metabolite floating around for us to characterize. And we incubated it under acidified conditions, about pH 5, in the presence of the reducing agent dithiothiritol, because the lysosomes are known for a reductive environment. We pulled a lot of time points. You can see some really early ones, and that becomes important later, and incubated out to 4 hours. And we included a positive control for cathepsin activity and a buffer-only sample. And you can see some of the baseline activities associated with these human liver lysosomes before we started the incubation. We took a fairly simple metabolite profiling method, and this was on purpose because we wanted to be able to do this for a lot of things, and we wanted it to be quite versatile. So you can see it's quite a simple gradient. It's only 5% to 95% over 11 minutes. The only really unusual and interesting thing about it compared to a traditional small molecule MetID method is that we are using a peptide BEH column. That's pretty important for the separation we wanted to achieve. And then we ran this on a QTOF using MSE mode, which means we have data-independent acquisition of precursor and product ions consistently. So at the end, you can mine the data for anything that you're interested in looking at. Speeds things up. And when we did that, we saw that in the lysosomal fraction, because the peptidase activity is so high, we saw extremely rapid loss, and it's actually double exponential. In some of the versions, in this one, it's single exponential, but it's extremely rapid loss. And you can see the half-lives are about a couple of minutes, which was the problem associated with GLP-1 in the first place. For us, it's okay because we're not trying to elicit any effects, but it does mean that it's super important to pay attention to the protein concentration. So from this point, instead of using 0.25, we moved into 0.05 mg per ml. And we also were including those very early time points because the activities here are so high. The whole job of a lysosome is to concentrate high enzyme activity to degrade things. We see the exact same effect in tirzepatide. Positive control, which is aminomethylcoumarin, established really nice cathepsin B activity in these samples, so we feel good about that. And then we established with our orforglipron that there was no real clearance for the small molecules. We don't have a lot of CYP activity or anything going on, which is good. So we didn't progress that compound for the rest of the study. And then we did some work where we used a workflow to find metabolites using a fairly broad search approach with an in silico digest. And then we also had a look for the fatty acid biotransformation metabolites, but that applies more to a different part of the work. But we kept the charge states and the isotopes pretty broad. We wanted to capture lots of things, whether we knew they were going to be there or not. And here is a picture of how we look at the data. So we do this in silico digest. We do have to add in custom amino acids and moieties. Fatty acid substituted lysine, for example, isn't going to be built into the software, so you have to add it in. And when we did that, we got some really nice metabolite profiles. We were actually tracking to make sure we could get all the way to the free lysine fatty acid, which you can see marked by the star. But we also saw a lot of different peptide cleavage products, all the way down to tripeptides. You can see in the earlier eluting portion of the chromatogram. Then we had a look at the MS-MS data. That's important, especially when you're looking at smaller metabolites of peptides, because you're going to have a lot of tripeptides or peptides with 4 or 5 amino acids that are endogenous. And so the sequence will help you determine if they're endogenous peptides, if you're really truly looking at metabolites. And so that's when the MS-MS data becomes useful. We identified 37 peptide cleavage metabolites, and you can see them here. And then we also were able to establish the free fatty acid modified lysine, which was important to us because it told us that we'd taken the reaction all the way to completion. We also looked a little bit at the linker and spacer metabolites because we did find them in lysosomes. We weren't sure if we would. And so these metabolites are formed by, on the left, amide hydrolysis at the linker, you can see right here, which makes sense because there are a lot of hydrolases in lysosomes, including amidases. And then on the right, we see dehydration of the free lysine fatty acid. We believe this is the aldehyde metabolite, but technically you can't actually specify 100% in the mass spec, so that's why it says that it's a water loss. So we established that some of those metabolites were occurring in the lysosomes. And then we took all of that information and we compared 4 different test systems: human liver lysosomes versus human liver S9 and under 2 conditions: acidified for the lysosomal piece and the catabolic enzymes, and then in more neutral pH to look at the extracellular space, be the cytosolic fraction, that sort of thing. And we compared it all to human hepatocytes as sort of our gold standard. And when we did that, we saw some very interesting things. If you compare A to B, you can see that the metabolism in the human liver S9 at pH 7.4 is much slower than in the human liver lysosomes, which is exactly what one would predict. If you then compare from A to C, you can see that the human liver S9 at pH 5 is much closer in terms of clearance rates to the lysosome data. And then the sandwich cultured human hepatocytes, which we actually did culture out to 7 days, have, again, much slower because it's closer to the in vivo clearance. You've got a lot more effects. There's uptake factors to come into play as well. We saw the same kind of thing, the same kind of pattern for the tirzepatide clearance. And it makes sense because a lot of people say, instead of using the lysosomal fraction, you can use a pH 5 S9 to mimic that. The problem is that although it appears to be accurate in the clearance data, it's not actually, the situation isn't exactly how it looks. So if you take it a little step further and you look at all the metabolites that form, you will see extreme differences across the test systems. I've highlighted here some of the areas in the peptide backbone where the differences are most marked. For the central endopeptidase activity, it's much less different across the subcellular fractions. But if you start looking towards either termini, then you will see some marked differences. And in fact, if you look at the lysosomes and compare them specifically to the pH 5 S9, you will see the pattern is not the same. The metabolism is not the same. The metabolism isn't the same in box 1, box 2, or box 3. So just because the clearance is matching doesn't mean that it's a true good mimic for the lysosomal fraction. We saw the exact same thing with the tirzepatide, same problem. And prior to some of the work we've done most recently, we were trying to establish why the differences were so marked. There are a couple of easy things you can say. You can say in the S9, the lysosomal enzymes are much more dilute than they would be in the S9. That makes sense. Than they would be in the lysosome, sorry. That makes sense. We know that. So maybe they're not as active because of that towards the metabolism of the drug. You can say, okay, we're going to manipulate the acid piece to find out if it's an acid-dependent enzyme or a neutral-dependent enzyme. And so we put together some hypotheses, sort of like how we used to back in the day when we didn't have as many recombinant enzymes for cholinesterases or carboxylesterases, and we would look at different differential tissue expression to say, okay, that one's carboxylesterase 1, that one's carboxylesterase 2. So we did that and we came up with some hypotheses, but unfortunately, we did this before the acidified data were available. When we added in the acidified data, the hypotheses didn't hold very well. Fortunately for us, we're in a period of scientific research where a lot more work has been done on looking at the lysosomal fraction and proteins in the in vitro test systems. So recently, there was a PRINCE consortium paper published, it was in May, looking specifically at hydrolases and transporters in human hepatocytes, S9, and lysosomal fraction. This provided some key insights for us that helped direct where we went next. The paper was actually based on characterizing some of these things in adults and children. It's a really good paper. If that's something of interest to you, go take a look. But it specifically identified 66 lysosomal hydrolases and 41 transporters. There were more proteins detected. That's just what was specifically identified and called out in the paper. And it turned out that the top 10 abundance proteins accounted for 66% of the lysosomal protein in hepatocytes. So the proteins in this list are clearly taking up an outsized contribution of the peptidases in these test systems. Cathepsins B and D alone accounted for 41%. So in collaboration with our collaborators that I will call out in a second, we were able to take the samples that we had, the various test systems, and perform some proteomics analysis, some very nice analysis actually, on an Orbitrap Astral with some nano-LC. And this uses another data-independent acquisition technique and some neural network software workflows to be able to derive an awful lot of proteins in the samples, and the data set's something like 7,500 proteins. If you would like more details on this, refer to the paper. And when we did that, we could compare the cathepsin protein abundance across the liver test systems. So in these two graphs, you can see on the left, that's the full scale, looking at everything all on one scale. And you can see there are 3 very high abundance cathepsins, B, D, and C. 10 of these are most abundant in the lysosomes of all the cathepsins that we looked at. 7 were present in lysosomes at about 3 to 5x the liver S9 abundance. And then 3 were at greater abundances than that compared to the liver S9. If you zoom in and you look closer at the low abundance proteins, you can see another interesting effect, which is that cathepsin G is most abundant in the hepatocytes. That's because it has a very high isoelectric point, and so it tends to bind electrostatically to the membrane during some of these processing steps. So it's lost when you look at the S9 and lysosomal fraction because you basically threw it away in the pellet. E and V were not observed in the liver test systems. Then if we look specifically at liver lysosomes versus liver S9, we can quantify that a little bit better. The B, D, and the Z were present at 3 to 5x higher abundance in the lysosomes versus the liver S9. If we look at the lower abundance proteins, things get a little bit more interesting. You can see that A, C, F, L, and S are more abundant in the lysosomes. They're about 2 to 5x the liver S9 abundance. But G is a little bit more abundant and fairly close, actually, to that liver lysosomes in liver S9. O has negligible abundance in everything, so we didn't look at that very much more. And so it's quite interesting because it helps you think about the fact that the protein expression in these fractions are completely different. The cathepsins are lower in abundance in liver S9 compared to lysosomes. We expect that because of the dilution factor. But if you look at the different proportions of the cathepsins, it is actually quite different between the liver S9 and the lysosomes. It isn't just that the proteins are present at the same abundances in the same contribution across cathepsins. But you can only really see that if you look at the low abundance proteins. So now we know the cathepsins are pretty different. We also looked at a subset of known lysosomal proteins, which I'm calling the non-cathepsin peptidase abundant proteins, and we looked at their abundance across the liver test systems. You can see that there are 3, CNDP2, PPT1, TPP1, which are present in much higher abundance than the rest of these peptidases. The CNDP2 is the highest in liver S9, which is the first one we've seen where that is a strong observation. Then PPT1 and TPP1 are highest in lysosomes. If you look closer and you look at the low abundance proteins, you will see that DNPP, DPP3, and TPP2 are higher abundance in the liver S9 versus the lysosomes. So it's inevitable that we will see different peptidase activity because there's more of different proteins. And then some of the others are highest in the liver lysosomes. Then if you could directly compare liver lysosomes versus S9, you can see that of the 3 highest abundance proteins, the CNDP2 is about 4x the lysosome abundance in the liver S9, whereas the PPT1 and TPP1 are much higher in lysosomes compared to liver S9. Whenever something was a little more than 5, I turned it red, so everything red has a bigger difference between the two test systems. If you look at the low abundance proteins, you can see that a couple of them are higher, actually 3 are higher in liver S9 compared to the lysosomes. And then a few of the others the rest of them are higher in lysosomes versus the liver S9. So these proteins are very, very different in terms of expression, abundance, and ratios. So overall, we were able to demonstrate the lysosomes as a practical option to look at catabolism of endocytosed peptides. We compared several different test systems for peptide metabolism, and we saw marked differences in the way that the biotransformation occurs in these test systems. Although acidified S9 demonstrates very similar clearance rates to lysosomes, the true metabolism and the observed biotransformations in liver S9 versus liver lysosomes are really quite different, and that could make a critical difference for specific situations depending on the question being answered. So in general, I would say we would want to use the lysosomes rather than using the acidified S9 mimic as a general rule. We did establish some non-cathepsin and cathepsin peptidase profiles in liver S9. They're quite different, especially if you look at the non-cathepsin peptidase profiles, which explains some of the differences in the metabolism that we saw. And we have also established overall the key takeaway here is that very considered experimental design and the right tools make a huge difference to the success of in vitro assays when you're looking at peptide metabolism. Moving forward, we are going to look a little bit more at some of the broader peptidases, not just the ones that we know to be enriched in lysosomes. And we're also evaluating rat trisomes as a model of the human lysosomes. I would like to acknowledge a few people that did the actual work here. I don't get to go in the lab very much anymore. I get to look at the data, but not so much hands-on with the instruments. Nadia, who did all of the biotransformations and structural elucidation, and Leah and Seema, who helped with the assays and with interpretation, and then Chris Boll, who's been a source of a wealth of knowledge about lysosomes. And then I would also like to acknowledge Bhagwat and Darshak at the Cincinnati Children's Hospital Medical Center for the really nice proteomics work, because they took care of that for us. And with that, I'm happy to take any questions, and Tina and I will be very pleased to share any insight we can with the rest of you.
Thank you for the presentation. It's really nice to see such a good application of quantitative proteomics that's close to our heart. So we have a couple of questions that I'll just read from the chat. So the first one says, will we expect differences between lysosomes from different organs? Because now targeting to different organs are becoming important. Thus, can lysosomes from different organs lead to different metabolism?
It's a great question, and it's something that's been quite prevalent in my mind of late, so it's timely for me. So right now, one of the challenges is we don't really know. We don't know that yet. If you look in literature, they don't look as different as one might expect. But I would say at this point, it's early days in trying to evaluate that. We have worked before with kidney lysosomes and a few other tissues. Some of the effects are very similar. The problem for us is because we look at so many different drugs and programs, it's hard to draw conclusions a little bit. But I think it's an area where we need more research in the longer run to be able to evaluate.
And then I have a question, actually. So given that cathepsins are so prevalent in the lysosome, what do you see as the implications for DDI in the lysosome? I mean, do you see any sort of drug-drug interactions happening in the lysosome, or is it just the elimination part that is happening there?
So I feel like in the lysosome, you actually have a bit of an advantage because there's so many enzymes and they're present at such high abundances that you're probably just going to see something else take over clearance. So it's not like you're going to inhibit clearance. I don't think. I mean, obviously this is speculation. There's only so far you can go in terms of experimental approaches. We have not specifically looked directly at that, and we tend to think of the drug-drug interactions on more of a cellular and tissue and full body level than subcellular fraction organelles. But it seems to me that if you inhibit, I know when you inhibit, like if you're looking across esterases or something, if you inhibit one very often, another one just takes over and you have no difference in the clearance. And I suspect that is what would happen with cathepsins. Tina, anything you want to add to that?
No, I think you covered it, and it's definitely your area of expertise, not mine. All good.
All right, so well, given that we are at the hour, I think I'm just going to thank our speakers one more time for giving such a great talk. And as far as our next webinar, is PQ's next webinar, we're going to take a break in July and then start up again in August. So look for more information from us then. And I hope that you all have a great day. Thanks again to Joanna and Tina for such a great presentation. Goodbye, everyone.
Thank you.
Thank you.
Mhm.
Bye.